We have 2 more talks to go. But I would like to thank you and introduce the next speaker of today's session. So our next speaker is Professor Kyle Orwig. I have to say that I have never met him in person, but I am old enough to remember him giving a fantastic presentation several years ago at one SSR meeting. He was probably still a trainee, and I remember how excellent his science was, and he has carried on this way for the last 2 decades at least. He is now professor of obstetric gynecology and reproductive science at the University of Pittsburgh School of Medicine, and is also the director of the Fertility Preservation Program at the Center of Reproduction and Treatment at Maggie Women's Hospital. He has been interested in and focused in his research on stem cell, especially spermatogonial stem cell and germ cells, and he's quite passionate about the translation of science to the clinic in order to diagnose, prevent, and treat infertility. And Professor Orwig, the floor is yours. You can share your screen whenever you're ready, and go ahead.
Okay, does that appear fine?
Yeah, that's perfect.
Okay, well, I'd like to start out by thanking Professor Calafell and the other organizers for the opportunity to speak here today. I'm only sorry I wasn't able to join you in person in Barcelona. I've really enjoyed the talks that I've seen so far. Well, the title of my talk today is Spermatogonial Cell or Testicular Tissue Transplantation for Restoring Fertility After Cancer Treatment. So, as you all know, medical treatments such as chemotherapy or radiation treatment for cancer, or medical treatments for other conditions like bone marrow transplantation or gender dysphoria, can cause permanent infertility. Adult men and women have the options to cryopreserve eggs, or cryopreserve sperm, or cryopreserve embryos before treatment. And these can be used to achieve a pregnancy in the future using established assisted reproductive technologies that we've all heard about during this research day. Unfortunately, those options are not available to prepubertal boys and girls, and that's simply because they're not yet producing mature eggs or mature sperm. And we think that this is an important human health concern because the good news is that the 5-year survival rate for kids with cancer is very high, now over 85%. But what that means is that we're now producing a lot of survivors who still have their entire reproductive life in front of them. And they may not be thinking about the family that they may want to have in the future while they're still young kids, but maybe that's something that we as a medical and research community can help them to think about. Well, as it relates to the boys, even though they're not yet producing sperm, they do have spermatogonial stem cells in their testes that are poised to begin producing sperm at the time of puberty. And there are centers all over the world now who are actively cryopreserving testicular tissues for boys, also ovarian tissues for girls, because we believe that in the future there will be technologies available to mature those tissues and produce eggs or sperm. Well, we established our fertility preservation program in Pittsburgh in 2010, and in 2011 we gained approval to begin cryopreserving testicular tissues for boys and ovarian tissues for girls or women, and since that time we have frozen tissues for over 400 boys and for 122 girls or women. We're proud of the fact that patients have traveled from all over the world to access experimental fertility preservation options that are available in Pittsburgh, which is indicated here by the yellow star. But this travel can be a barrier in access to care because, as you can imagine, the cost of travel may be a problem. and or the family may have a sick child who simply isn't able to travel to Pittsburgh. So to address that problem several years ago, we established what we call a coordinated network of centers. And with this model, so the coordinated network of centers is indicated by all of the green stars and blue stars. And in this model, a patient can have a testicular biopsy or an ovarectomy at the same institution where they're being treated for their primary disease. And with this model, a patient who's being seen in Phoenix can have their tissues sent to the Yellow Star in Pittsburgh instead of the patient traveling themselves. And of course, in Pittsburgh, we have the infrastructure and the expertise to process and to freeze those tissues. Well, when young patients come to us asking for fertility preservation, this pie chart indicates the kind of diagnoses that they have. And the ones that are prevalent in kids include sarcomas, brain tumors, leukemias, and we have over 20% of patients who are getting a bone marrow transplant for benign diseases such as sickle cell anemia or beta thalassemia. And increasingly, we're beginning to see patients who have gender dysphoria and want to cryopreserve their reproductive tissues before they initiate gender-affirming treatments. Well, when we receive tissues in Pittsburgh, either from our local institutions or from one of the coordinated centers, the tissue processing is quite simple. We cut the testicular tissue biopsy into small pieces that measure between 2 and 5 millimeters across. And then we use controlled slow rate freezing to cryopreserve the tissue. So to review, the standard and established fertility preservation method for teenage or adult men is to cryopreserve a semen sample with sperm, because we know that those samples can be thawed in the future and then used to establish a pregnancy using existing assisted reproductive technologies. That option, of course, is not available for prepubertal boys because they're not yet producing sperm. And so I've told you about the option of cryopreserving testicular tissues. And we believe that those tissues can be used in the future and matured to produce sperm using either cell-based therapies or tissue-based therapies. And today I'm going to tell you about two developing technologies that I believe are quite mature and may be ready for translation to the human clinic. And those include spermatogonial stem cell transplantation and autologous testicular tissue grafting. So I first want to tell you about a study that we conducted several years ago in what we call the monkey model of cancer survivorship. This was work that was led by Brian Herman when he was a postdoctoral fellow in the laboratory. He's now an associate professor at the University of Texas in San Antonio. So Brian treated rhesus macaques with an alkylating chemotherapy called busulfan that we knew would cause infertility. Before he did that, he took a biopsy from the testes of those animals and cryopreserved a testicular cell suspension that came from that biopsy. He then treated the animals with chemotherapy and every week thereafter collected a semen sample to determine the impact of the chemotherapy treatment on future fertility. And the results of that early experiment are shown here. Animals were treated with 3 different doses of busulfan. And what we found is that compared to the control animals, which are shown in green, all doses of chemotherapy caused sperm counts to go down to 0 or near to 0. Now, the animals that were treated with the lowest dose, shown in the red line, you can see that sperm counts eventually came back up into the normal range. And this is probably because at least a portion of the spermatogonial stem cell pool survived the chemotherapy insult and over time began to regenerate spermatogenesis. But the animals that received the higher doses of chemotherapy, shown in the yellow and the blue line, you can see that sperm count stayed at 0 out beyond a year. And we figure that that represents the 30% of patients who might be rendered permanently infertile as a result of their chemotherapy treatment. And this is probably because the stem cell pool was completely depleted by the chemotherapy treatment. So Next, Brian asked whether it would be possible to thaw out the cryopreserved testicular cell suspension and transplant it back into the testes of the animal from which the tissue was obtained originally. Before he did that, he treated the cells with a lentivirus that carried the green fluorescent protein gene. It turns out that the green fluorescent protein is not a good diagnostic in monkey semen or monkey sperm because monkey sperm glow green anyway, but the lentivirus has the unique feature that it integrates its genome into the host cell genome, so we can use PCR to determine the presence of lentivirus, and this will tell us unequivocally what was the fate of the frozen, thawed, and transplanted cells. So the next question was how to introduce those cells into the testes. So for this, we followed on techniques that already had been developed in pigs and sheep and goats, and that's a technique called ultrasound-guided Grady testis injection. So what you're looking at here is an ultrasound image of a testis. It turns out that the Grady testis is easy to visualize on ultrasound because it is this echo-dense white line, and the Grady testis is connected with all seminiferous tubules of the testis where spermatogenesis occurs. So the technique involves simply inserting a 25 gauge needle through the base of the scrotum until the tip of the needle emerges into the Grady testis space. We put in microbubbles in the cell suspension because those are visible on ultrasound, and this allows us to track the progress of the transplant. So first you see the Grady testis fills from end to end, and now you can begin to see the seminiferous tubules all filling up out of the Grady testis. So here are the results from one animal on that study that received an autologous testicular stem cell transplantation. You can see that this animal was producing sperm at the beginning of the study. We treated him with the busulfan chemotherapy, and about 10 weeks later, sperm counts went down to 0. So at that time, we thawed out his testicular cell suspension and transplanted it back into his testes. And here you can see that about 30 weeks later, sperm counts began to rise into the normal range for a peripubertal rhesus macaque. And we know that at least some of those sperm came from the frozen, thawed, and transplanted cells because when we did PCR for the lentivirus and GFP gene, we could see a signal for that gene in the ejaculated sperm. Of course, that signal was not present prior to transplantation. I won't have time to show you the data today, but we did go on to examine whether these sperm were functionally competent to fertilize rhesus oocytes, and they were. These sperm could fertilize rhesus oocytes, and they led to pre-implantation embryo development up to the blastocyst stage. We still need to show that we can establish a pregnancy and produce a healthy offspring that way, and that is the focus of our ongoing experiments. So I hope I've convinced you that it is at least technically possible to transplant spermatogonial stem cells into the testes of a chemotherapy treated recipient and to regenerate sperm production that way. I believe that we will be able to translate this technology to human patients because here you're looking at the ultrasound images of the testes of 2 teenage boys, and I think you can appreciate, like I do, that the Grady testis is easy to visualize here, just like it was in the non-human primates. So with that, I'd like to suggest that spermatogonial stem cell transplantation is ready for the clinic today. And that's because this is a mature technology that was originally described over 25 years ago by Ralph Brunster and colleagues. He initially demonstrated and proved the technique in mice, and that technique now has been replicated by other groups in mice, rats, hamsters, dogs, cats, goats, sheep, pigs, and monkeys, with the production of healthy offspring in mice, rats, goats, and sheep. Also, I believe it's important to bring next generation technologies to the clinic because we can document in published reports that testicular tissues have already been cryopreserved for more than 1,000 patients worldwide, including the 401 patients at our institution. This is certainly an underestimate. I'm confident that there are quite a few more tissues than that have been frozen. And since these patients have already undergone and taken on the risk of a surgical procedure to preserve the possibility of future fertility, I think we owe it to those patients to responsibly bring next generation technologies to the clinic. The other thing that's not widely known is that spermatogonial stem cell transplantation actually has already happened in the human clinic. This happened in a study by Radford and colleagues in 1999, where testicular cell suspensions were frozen for 12 patients with Hodgkin's disease. We later learned that 7 of those patients returned to have their cells transplanted back into their testes. We never learned anything about the outcomes of those experiments, and the technology was in very early stages in those days. It only had been done in mice and rats at that point. Well, even though we don't know the outcomes, I do think that these studies tell us something important about the motivation of patients who are willing to undergo an experimental and early stage therapy for at least the possibility of having a biologically related child. So, if we were going to take spermatogonial stem cell transplantation to the clinic today, who would be the ideal patients and what age would be the best time to do the transplantation? And what I'd like to suggest is that the best patients would be patients who either have solid tumors that aren't known to metastasize to the testes, like a sarcoma, for example, or patients who are getting bone marrow transplantation for a benign disease like beta thalassemia or sickle cell anemia, because in these cases there is limited or no risk of transplanting malignant cells back into the patient. And regarding the time to do transplant, based on 25 years of data in animal models, I believe the best time to do transplant would be during puberty, because at this time, under the influence of gonadotropic hormones, the testes are growing, and this probably creates more niches for the stem cells to engraft and to regenerate spermatogenesis. Okay, so for the rest of my time, I'd like to tell you about a tissue-based technology that I also believe is mature and ready for translation to the human clinic, and that's autologous testicular tissue grafting. So again, we return to our monkey model of cancer survivorship. This was work that was led by Adetunji Bayomi when he was a graduate student in the lab, and Karen Peters, who was a research associate, and now she's a nurse actually on a cancer floor at our institution. So again, we collected a biopsy from the testes of animals that were going to get treated with chemotherapy. In this case, instead of freezing a cell suspension, we froze intact pieces of testicular tissue. Having preserved the fertility of these animals, we then treated them with chemotherapy, and then after the chemotherapy treatment, the tissues were thawed and transplanted under the skin of the same animal. So first I want to show you the histology of these testicular samples so that you can understand that these are immature testicular samples. Here you're looking at a cross-section of the seminiferous tubule of a testis. You can see that it contains a small number of VASA-positive red germ cells, but because this is from an immature animal, they have not yet organized into a seminiferous epithelium. And of course, there are no Akerson-positive green postmeiotic spermatids or sperm. So we used those immature tissues in this experimental design where the animals were treated with chemotherapy. In this particular experiment, we also castrated the animals so that we can unequivocally determine the outcomes from the graft and distinguish those from endogenous testis outcomes. Of course, we will not castrate prepubertal boys before they undergo a cancer treatment. And for each one of the graft sites, we individually sutured 4 pieces of tissue to the underside of a skin flap, and then we simply sutured the skin flap shut. The experimental design was that we grafted tissues into 6 locations under the back skin and 2 locations under the scrotal skin, in a design that allowed us to compare graft outcomes in frozen and thawed tissues compared with fresh tissues. Of course, we're most interested in the frozen and thawed tissues because that will be the scenario for our prepubertal cancer patients. This design also allowed us to compare graft outcomes under the back skin versus under the scrotal skin. And we injected Matrigel into some of the graft sites because we thought that that might stimulate angiogenesis into the grafted tissue. Well, we got an early indication that the grafts were functioning because after several months, we began to see testosterone levels rise up into the normal range for a peripubertal rhesus macaque. And we know that this testosterone had to come from the grafted tissue because remember, these animals were castrated, so there was no endogenous source of testosterone. And over time, we began to see those small pieces of grafted tissue grow into large lumps under the back skin or under the scrotal skin. And it didn't matter whether the grafts were frozen and thawed or whether they were fresh. It didn't matter whether or not we added Matrigel. And it didn't matter whether the grafts were under the back skin or under the scrotum. All grafts grew continuously throughout the duration of the experiment. And when we recovered those grafts 8 to 12 months after transplantation, you can see that the 4 little pieces of tissue have now grown and merged into relatively large masses. And when we looked at these tissues histologically, I hope you can appreciate that those immature tissues have now matured so that the seminiferous tubule now contains multiple layers of VASA-positive red germ cells and a wide open lumen in the middle of the seminiferous tubules. And there are numerous post-meiotic, acrosin-positive green spermatids and sperm. And when we mechanically dissected those tissues, we were able to release testicular sperm. And there wasn't just a few sperm. We were able to recover between thousands and millions of sperm from every single graft that was recovered. So the next question for us was, were those sperm functionally competent to fertilize rhesus oocytes and to establish a pregnancy? So to address this question, we reached out to our friends at the Oregon National Primate Research Center, who have a well-established monkey ART laboratory. We sent testicular sperm to them in November of 2017, and they fertilized rhesus oocytes using intracytoplasmic sperm injection. And we were able to learn that those sperm were competent to fertilize because ICSI led to the development of 2-cell embryos, 4-cell embryos, 8-cell embryos, and etc., all the way up to the stage of a hatching blastocyst, which is the stage when the embryo would implant in the uterus. So they next actually did an embryo transfer. This happened in November of 2017. We were able to confirm a pregnancy in December of 2017. 1 month later, in January of 2018, we confirmed by ultrasound that fetal development was progressing normally. So at that time, we scheduled a full-term C-section. And the result was the birth of Grady. That stands for graft-derived baby. She was born on April 16th of 2018, and she's the first monkey baby born from autologously grafted, frozen and thawed prepubertal testicular tissue, which we believe represents that model of a prepubertal cancer survivor. And like spermatogonial stem cell transplantation, we believe that this is a mature technology that's ready for the clinic. Again, this was originally introduced in mice, and it's a technology that's been replicated in numerous mammalian species, including now in non-human primates. And so I think that that establishes that this is a technique that's safe and feasible. And of course, the next animal model is always the human patient. So Grady still lives at the Oregon National Primate Research Center, and the value of that is that we get to monitor her development and her social interactions with other animals and her behaviors throughout her life. She is evaluated several times each year, and this is a video of one of those evaluation sessions. You can see Grady, she's hanging on the wall, and her surrogate mother is in the pen with her. So by all accounts, Grady is developing normally, and as I said, we will continue to monitor her development through to adulthood, and of course at that time we'll also be interested in assessing her fertility. So with that, I tried to acknowledge the people who did the work as I went through the presentation, but of course I've described to you 15 years worth of research, and many, many hands were involved in what was a very multidisciplinary set of studies. Also, we have collaborators all around the world, but I'd particularly like to acknowledge Stefan Schlatt and Ina Dobrinsky, who were the scientists who pioneered the testicular tissue grafting procedures, and were very generous in sharing their knowledge with us as we developed up our monkey experiments. I also want to acknowledge the staff in our UPMC McGee Center for Reproduction and Transplantation. This is the team that recruits patients and processes and freezes tissues for us. And finally, none of this would be possible without generous funding from a variety of sources. And I always like to finish my talks by acknowledging patients who inspire the work that we do, and in many cases donate their tissues to research to help us develop next generation technologies. So I'll finish up by showing you that this is what our lab looks like in 2020 and 2021. I hope very soon that we'll be able to join the lab without wearing face masks every day. And also, I hope sometime in the future I can come and visit everyone in Europe, and specifically in Barcelona. So as a last thought, I want to tell you about a Gordon Research Conference that will be held in the summer of 2022. This is in August, and it'll be held at Mount Snow in West Dover, Vermont. This was a meeting that was actually held in Tuscany in 2018. And one of the benefits of doing that is that the European participation was very high in that meeting. I hope we also can have outstanding European participation at the next meeting in Vermont. And I expect that the 2024 meeting will be back in Europe, and so that we can increase the international flavor of this particular meeting on mammalian reproduction. So with that, thank you for your attention, and I'll be happy to take any questions.
Thank you very much, Professor Orwig. And in fact, we do have already some questions. So one question is, do you know what is the damage that chemotherapy may exert on the niche that would support the transplanted spermatogonia?
Yeah, so that's a good question. And I cannot say that there is no damage of chemotherapy, but what I can say is that we've been transplanting into chemotherapy-treated animals for many, many years, and the environment is at least sufficiently functional to support complete spermatogenesis. And also, boys who are treated with high-dose chemotherapies usually progress through puberty normally without the need of exogenous hormones. Now, the situation is different for radiation. High-dose radiations can damage the somatic environment of the testis, and in some cases, those boys will need hormone supplementation in order to progress through puberty.
And then there is another question, it's a little bit along the same lines. So do you know if the procedure per se, the transplantation and the grafting, is mutagenic, if you've checked these cells before and after?
We haven't assessed that at a cellular level, but what we can say is that in all species that this has been tested, it leads to the birth of healthy offspring, to the extent that that's been studied. Of course, we don't study the overall health of mice and other animals in great detail, but as I said, in the case of Grady, we will have the opportunity to follow up. So that doesn't answer the question at a cellular level, but I think it begins to answer the question at an organismal level.
Perfect. I have a couple of questions myself. So one of them is, if you can use hormonal supplementation when you transplant a spermatogonial stem cell, so in a sense you can support the niche if it has been damaged, what would be the indication to transfer tissue then?
Yeah. Are you saying if you could regenerate spermatogenesis from endogenous, then there would be no need to transplant tissue? Is that your question?
No. I'm thinking about the two techniques: the transplant only the SSCs or transplant physical tissue. So my question is, if I imagine thinking about a patient with cancer, that one would go for the least risk of, to reintroduce the disease, right? So if you
Yes,
can,
of course.
if SSCs are enough, so to speak, with some support, why go into the tissue?
Yeah, so you're absolutely right. If you transplanted an autologous tissue, let's say from a leukemia patient or a testicular cancer patient, there would be a risk of putting cancer cells back in. I think what you're suggesting, that if we did a cell suspension, we may be able to separate the therapeutic stem cells away from malignant cells. We've actually done that study both in monkeys and in human testicular tissues by contaminating with a very malignant leukemia cell line. And indeed, we showed that we could separate the therapeutic stem cells apart. However, I would not be comfortable doing that, especially in the first cases, because I don't think we ever can be 100% sure, in spite of the data we published. I don't think that we can ever be 100% sure that we eliminated all of the malignant cells. And of course, reintroducing a cancer back into a survivor would be the worst thing that we could do, not only for the patient, but actually for the field. And so one day we may get there and we may agree as a broader community that it's a reasonable thing to do, but I think in the early stages, we should be very cautious about that.
And then another question is, you said it, in fact, more than once and twice, that the technique is ready for the patient, right? The technique is ready, the patient are ready. So are you waiting for someone to want a child?
Well, so we actually are approved now to do both spermatogonial stem cell transplantation and testicular tissue grafting in patients. And so we're actively reviewing the 400 patients that we have to identify who we believe will be best candidates for transplant. It's probably not obvious to everyone, but of course all of our patients are kids, or almost all of our patients are kids, and so many of them are still quite young. But we do have patients who are late teenage years or early 20s, and those would be among the patients who I think would be good candidates. And as I said, I think the best time to do transplant is actually during the teenage years and or during puberty, rather than waiting till our patients are ready to start a family. If we wait until starting family, we still may wait another 10 years because that's just the state of the technology and the age of our patients.
Perfect. And I would just like to conclude with one question. So you show, especially with monkey, this incredible work, which it's incredible, but it's N of 1, right? So
yes.
Do you have an idea in primates what is the efficiency? Have you tried in a group of animals, and does it always work, or does it often work?
So the transplant technique itself, I've done in over 100 monkeys. The reproducibility in terms of regenerating spermatogenesis is excellent. And it's not just in monkeys, of course, this has been replicated in numerous animal species. So the N is very, very high. Now, the N of producing healthy offspring in monkeys is very, very low. It's 1. And for financial reasons, it would be nearly impossible to replicate this at a level that you could make statistical statements about. We haven't reported it yet, but we do actually have one more baby that was born as a male, born from grafted tissues. It was a different experiment. The tissues were actually xenografted into mice. And so I think over time we'll slowly begin to accumulate greater than an N of 1, but unlikely to generate a sufficient N to have statistical strength.
Perfect. Well, this is the way it goes. I mean, with the ovarian cryopreservation, eventually we move to patient, and little by little, you may experience and numbers, and until when you realize it's not experimental anymore.
Yeah, that's exactly right.
Okay. Well, I would like to thank you, Professor Orwig, for this talk and this very nice conversation. And thank you again for being here with us, waiting for in-person meeting in Barcelona. And I think we can move on to the next and last speaker.